The content provided in this podcast is for informational and educational purposes only and is not intended as medical advice, diagnosis, or treatment. Always seek the advice of your physician or other qualified healthcare provider with any questions you may have regarding a medical condition.
Is it my story, your story, his story, her story, Jane's story, Joe's story? Is it just an allegory? Show's about to start and we still don't have a name for it. Pop, 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 pop. It's a long story.
It's a long story.
Wow, that was deep and it lingered. It was
Amazing.
awesome.
It was
Thank
deep.
you. Great opening. And it's a great opening because today's episode is a really fascinating one. It's about pivoting back in time where the enemy of your enemy becomes your friend. And when antibiotics stop working as well, we turn now to viruses. But before we unleash today's very targeted conversation with a couple of incredible guests to talk about two perspectives on the most abundant organism in the world, bacteriophages. I used to always say phages, but that's just not right. It's bacteriophage. It is my pleasure to first introduce the team that keeps this podcast functioning. First, my co-host Lauren, who is currently managing her own highly complex ecosystem. This podcast topic seems to be far less challenging than her upcoming August wedding. And we all know both require careful planning, precise targeting, and a backup strategy when you come against some resistance. But she doesn't panic. She strategizes. She adapts. Whether that's a phage or a seating chart. Welcome, Lauren.
Hello. I would say that's mostly accurate. I do panic, though. I definitely am not good at planning things, so I'm just expecting something's going to go wrong. I've also never been described as my own ecosystem, so I'm going to have to start using that. I don't think I've ever been described that way.
Well, we tried to bring in the phage ecosystem and the ecosystem of bacterial world into that introduction, but I may not have pulled it off very well.
I like it.
But
no, I'm going to start using that.
Are you like facing resistance, or you just like go with the flow? Everyone else is just handling it. Thinks that's what you're doing.
A little bit of both. Go with the flow, but apparently I'm very opinionated, so that has caused some issues. And my fiance is also very opinionated, so it's like we have two brides.
That.
He won't listen to this probably, so he'll never know I said that.
You won't know. And that's not the worst thing you've said. No, I'm just kidding. And then again, previously we had Andrea sitting in line or standing in line for the Mandalorian, but now she's taking a pause. And essentially, she is the bacteriophage of our operation. She identifies the noise, attaches with precision, injects just the right solutions, and suddenly everything works better. And with her growing marketing expertise, she's ensuring this podcast just doesn't inform, but it spreads intentionally. And Andrea, welcome on this. May the 4th be with you.
And may it be with you as well.
So we're recording today on May 4th. And so people won't hear this until June or at the end of May, but it's a big day for us. And behind me, I have a stormtrooper. And I think everybody on the podcast will be revealing their Star Wars fandom.
Item of choice.
Just a quick introduction to our podcast. And it's going to be less than a minute here, little history for everybody. But in 1917, a French-Canadian microbiologist named Félix Durel, although I did find Frederick Trott, and I don't know if they're pronouncing their names correctly, we'll ask our guests, but they discovered tiny viruses that devoured bacteria, and they called them bacteriophages. And my son will appreciate this, who is a classics major, but it's from the Greek to eat. He was so confident of this discovery, he drank the phage cocktail himself to show that it was safe. And then he gave it to a 12-year-old boy who was saved from near death, suffering from severe dysentery. He made a full recovery. And the idea of that captured the world's imagination. In 1925, an author by the name of Sinclair Lewis wrote something called The Arrowsmith. It was a novel about a young doctor who used bacteriophage to fight the plague. And later, he won a Pulitzer Prize, but also the Nobel Prize in Literature for his work that included that. So it remains one of the most celebrated novels in the history of medicine. And then came penicillin. So Jerry Nick, he's a pulmonologist that is a colleague from National Jewish in Denver. He eloquently put it that penicillin and the Allies beat the Nazis and fascism. Antibiotics, he said, became the miracle drug during the 20th century, and the West abandoned phages and almost overnight. And meanwhile, behind the Iron Curtain in the Soviet Union, particularly Georgia and Poland, kept using bacteriophages, and quietly they've been working on this, and they've been now they're the center of excellence ever since. Now, a century later, we've come full circle. Antibiotic resistance is claiming nearly 5 million lives a year. And those ancient viral warriors, the bacteriophages, are surging back into the mainstream. And so it is truly a privilege to welcome our next guest, Dr. Stephanie Strathdy, a globally recognized infectious disease epidemiologist, visionary leader in bacteriophage therapy, and building the scientific and regulatory frameworks needed to bring this precision therapy reality worldwide, and one of the most influential voices in the fight against antimicrobial resistance. Dr. Strathdy is the Harold Simon Distinguished Professor of Medicine at the University of California, San Diego, where she co-directs the Center for Innovative Phage Applications and Therapeutics, the first dedicated phage therapy center in North America, as well as the international core of UCSD's Center for AIDS Research. Through her work, she has helped bring phage therapy into the mainstream conversation about how we treat multi-drug resistant infections, and her work spans infectious diseases, global health, and implementation science, with more than 700 peer-reviewed publications and recognition as one of Time Magazine's most influential people in healthcare. I've heard her talk numerous times, including an episode of Radiolab recently, and we invited her today to share her journey into this field and how, after her husband Tom developed a life-threatening infection with a multi-drug-resistant bacterium, she helped orchestrate one of the first successful uses of intravenous bacteriophage therapy in the U.S., an effort that not only saved his life, but helped catalyze global resurgence of interest in phage therapy, which you can also read about as Steph and Tom co-authored a memoir about their experience called The Perfect Predator. So to get us started, Dr. Strathdy, can you start us off by taking us back and sharing a brief version of your story, including the moment when you first Googled alternatives to antibiotics?
Absolutely. And I just want to first thank you for inviting me on this show. I really feel close to the CF community because I've come to know many patients, some who are with us and some who are no longer with us. And, I feel like I'm part of the community too. And of course, like I didn't plan on becoming this figurehead in the phage therapy field. I fell into it. My husband and I were on vacation in Egypt in 2015, and he looked to be in good health, except he was overweight. So one moment he was crawling through pyramids, and then the next moment he was fighting for his life. And there was no hospital in Luxor where we were based. So they stabilized him as best they could, and we got a medevac first to Germany and then back to San Diego where we were based. And we learned along the way that he had a gallstone that had stuck in his bile duct and caused an abscess to form, and he'd been walking around like that without knowing it. But along the way in Egypt, he acquired a superbug infection, and a superbug is essentially a bacteria that is resistant to multiple antibiotics. And the bad thing is that in his case, it was one of the most deadly bacterial pathogens on the planet, and it became resistant to all available antibiotics. And so I felt blindsided because I'm an infectious disease epidemiologist. I'm certainly not a physician. I have a PhD and a rusty old degree in microbiology from the University of Toronto. But, all of a sudden, like this superbug crisis, it jumped up on me. I just didn't realize how big of a problem it was. And I was quite embarrassed about that. But, my husband became a poster child for this post antibiotic era that we're in right now because antibiotics have been overused and misused and they're no longer working. And patients with CF who are getting like lung infections and other types of infections who end up receiving lots of antibiotics end up getting bacterial infections that become resistant to the antibiotics that are available. And so that's why this is such an important topic for this community. So in my husband's case, there were no antibiotics left. They couldn't operate because they felt that the bacteria would get into his bloodstream and he would die. And he was slipping away a little by little each day. And I had a conversation with him where I asked him if he wanted to live. It was just horrible. He was in a coma, and I said, that I wanted to grow old with him, and I knew he was really tired, and if he wanted to leave, I would understand, but if he wanted to live, please squeeze my hand, and he did. And so I got really excited, but then I got freaked out because I realized, I didn't really know what I was doing. And some of the top infectious disease physicians had been treating him and they'd run out of solutions. So I did my own research and I stumbled across this bacteriophage therapy. And you can say bacteriophage if you're French, like phage is how you would say it.
Thank
Phage,
you.
phage. So in English, we shorten it to phage, and phage are viruses that have naturally evolved to attack bacteria. So, just like, the lion is the predator that, eats a gazelle, right? Like the bacteriophage eat bacteria, and it's very specific to bacteria. So bacteriophage don't attack human cells, only bacterial cells, and they only attack the bacteria that has a receptor that they can connect to, and they drill into the bacterium and take it over and turn it into a phage manufacturing plant. So it's almost like the bacteria is like a zombie, right? It's not making bacteria anymore, it's making phage. And when given the kill signal, the bacteriophage that are baby phages inside burst out and kill that bacteria in a process called lysis, and all those baby phages then go on to attack new bacteria. Again, only the bacteria that have the receptor that they can attach to. So it's precision medicine at a nanoscale. I thought, wow, like, why aren't we doing this? And I dug a little deeper and realized, well, Western world had forgotten all about phage because they had embraced penicillin and all the antibiotics that came after it. And it was only in places in the former Soviet Union that were, still using phage. So the research that showed that it worked had never really been done. So it was considered experimental treatment, and my colleagues at UC San Diego agreed that if we could find phages that were a match for Tom's bacteria, that they would ask the FDA to provide approval on a compassionate basis because he was going to die. So that's what happened, and we reached out to total strangers, and they were phage researchers, especially at the Texas A&M University. They put a phage cocktail together. They sourced phages from, sewage and duck ponds and barnyard waste. It was because wherever you find a lot of bacteria, you find the perfect predator, the phage, that prey upon them. And even the U.S. Navy was approached and got involved because they had been collecting phages from the bilges of ships around the world. And they'd never used them on people before. And they were excited to try it. One of the reasons they agreed to treat a civilian is because they realized that this could jumpstart the field 10 years or more. So basically my, husband was a giant guinea pig, as he describes himself now. He was so close to death, and when we injected these phages into him, it was a billion phages every 2 hours for a couple of days, and he lifted his head up the pillow and woke from a deep coma and kissed his daughter's hand. So everybody in the ICU freaked out. There was, like, happy tears all around. He made as close to a complete recovery as you can expect after somebody like him lost 100 pounds and had to learn how to talk and walk and swallow and do everything again. He was like a giant baby. He'd been in the hospital for 9 months. 4.5 months was in the ICU. And he came home and has recovered enough that he went back to work and is doing great. But, I just went into this wanting to save my husband's life. I never dreamed that this would be seen as a forgotten cure that could help other people. But as soon as we realized that other people were using the protocol that we used on Tom and it was saving lives and limbs, then, we decided to write a book, The Perfect Predator, to share our story and to educate people about the superbug crisis and the promise that phage therapy holds so that we can hopefully expand it and get the research needed to show that it works so that it can get approved alongside antibiotics. So since this time, our team opened up the Center for Innovative Phage Applications and Therapeutics in San Diego. It's a nonprofit based at the University of California, San Diego. It became the first dedicated phage therapy center in North America, and now there's several others, and there's lots around the world. So phage really is all the rage now, guys.
That's amazing, and there's, so many things to pivot here. I just wanted to say a couple things. First of all, like, the movie Alien is playing out in a nanoparticle level, and so these bacteria, like, are fearing for their lives. And I think I read somewhere that bacteriophage eat about 40% of the world's bacteria every day, which seems crazy. Just, I guess that's how they all replicate so quickly. And also, I was going to say this: your Tom's story really resonates, I think, with the CF community. And I want us to come back to this because reading your book and reading the emotions, reading how as a caregiver, all the things you needed to do to fight, I think that resonates. And so I would love for us to come back to that. But as we're just learning about phage therapy, I just wanted to ask one question. First, how do we get phage out of the sewers and ready to deliver it to humans? You described, in bowels of ships, in the sewers, but how does that get to a person and ready for use? Like, what are like the steps in like just kind of as simple way as possible?
Sure. Well, I mean, phages are the most populous organism on the planet, and they're 100 times smaller than bacteria. It's just that we haven't realized the important role that they play in the microbiome, right? They're the gatekeepers keeping the number of bacteria in check. So they are in nature. They're in soil, they're in water, they're under rocks, they're in sewage. Wherever you find a lot of bacteria, you find a lot of phage. So it's actually very easy to isolate phage. And there's a program called the C-Phages program, headquartered at the University of Pittsburgh, that has chapters all over the world where students learn how to isolate phage. And those phage go into a library and the students that identify new phages learn how to sequence them and annotate them and even get to name them. And some of those phages have been used on CF patients. In fact, the very first time that a genetically modified phage was used to treat a human bacterial infection was on a CF patient in the UK, and it was successful, and it was just a landmark case. But phage basically, if you were going to do this, you get a sample of say sewage or soil or whatever, and you make sure that you can, have a liquid component to it. So you can, if it's soil, you would add some water to it, and then if the bacteria that you want to kill, say you streak it on a petri dish. And you incubate it so that it's growing, and you can see the bacterial colonies, and then you put the sample, of the sewage or what have you on the Petri dish, and you can incubate it for 24 to 48 hours at body temperature. And if it comes back looking like Swiss cheese, there are literal holes in the agar, in the Petri plate. And that means that you've got a phage that's gobbling up those bacterial colonies, and you get excited. So even though you can't see phage with the naked eye, you know that they're at work. And so then what you do is you can use a pipette to extract that phage from the agar plate, and you can add more bacterial suspension so that you're growing up enough phage to be able to treat the patient, because you need billions and billions of phage over time to treat a patient. So then the next step becomes purifying that suspension, and that's the tricky part, because when bacteria die in the process of being attacked by bacteriophage, the bacterial cell wall, especially if it's gram negative bacteria, because there's gram negative and gram positive bacteria, gram negative bacteria are particularly problematic, and their bacterial cell wall has something called LPS. And for those of you that are science nerds like me, that's lipopolysaccharide, and it's toxic to people. So you don't want very much of that, if any, LPS in the phage preparation. So you've got to remove it somehow. So there are ways to do that, but that's where things get complicated, because the FDA in the US wants you to measure how much of this potential toxin is in the preparation to make sure that it's safe to give to the patient. Right now, because phage is an experimental treatment in the US and most parts of the world, you have to ensure that you can reach labs that are going to do this kind of work. And most of these
Yeah.
labs are volunteer labs at universities, and they're not equipped to manufacture phage in sufficient quantities and remove the toxin, etc. So we often need to ship around like the phage preparation to different places to make this happen. So that's kind of the scalable issues that we need to overcome in order to take phage therapy to, a global level. It's all surmountable. It's just that we don't have the resources to do this. These days, the National Institute of Health and the National Science Foundation, which fund most of the research in this area and others, are being gutted. The current federal administration has really chopped budgets and people so drastically that even before this government came in, we were, really behind an 8 ball without the equipment and the facilities to be able to do this. And now it's worse. So those are some of the challenges,
So,
and we
so
we need clinical trials to be able to compare the phage against antibiotics, and so that all of that takes time.
So the sample is cleansed and scrubbed, but in a way that it doesn't harm the bacteriophage, but gets rid of the LPS. And this isn't something that anyone can just do in their backyard. I mean, it can be done in a lab, but not at a scalable way. And so we need that infrastructure and help from industry to sort of make this
Yeah, and
commercialize
we and
this a
we
little
also
more.
need advocates for phage therapy. One of the
Yeah.
most powerful advocates is one that you had on a former show. That's Diane Shader Smith. She was the mother of Mallory Smith, who wrote her memoir "Salt in My Soul: An Unfinished Life." And I was one of many people that tried to get phage to Mallory in time to save her life, and we were too late. But we came close, and, even though she didn't get phage in sufficient quantity and it wasn't, scrubbed to the level that we wanted, her parents decided to give it to her anyway, and she was already dying, essentially, and so they had to withdraw life support, but they decided to biopsy her lung after she passed, and the phage had already reached her lungs, and that's what convinced Diane that if Mallory had received phage therapy earlier, or what her dad wanted to do is give her phage prior to her lung transplant to wipe out the bacteria that was trying to, take her down, that
Yeah.
maybe she could have been saved. So that was the first CF patient that I got involved with. I also got to know the patient in the United Kingdom that received the genetically modified phage, and she had an extra 3.5 years of life before she succumbed to CF. She didn't die from her bacterial infection. She ended up going back to school and getting a job and going on vacation outside of her country for the very first time. So, I've seen a lot of success stories, and I understand that we have another phage therapy patient who's joined you today. So I'm excited to have this opportunity.
And Mallory, I mean, she had CF, and Diane joined us on a webcast that we had before this podcast became a reality. But yeah, really incredible, and you really did open the eyes of the CF community to this therapy. And with her, and then with Marianne O'Hara, one of Mallory's friends, also big advocate. So thank you for starting that, getting that ball rolling. I think it's maybe a good time for us to introduce Maggie into the conversation. Maggie Donovan, she received Phage in 2023, and we'd like to hear about your experience with that. But she is 23. She was born in May. May is CF Awareness Month. Her middle name is Rose, and the rose is the CF flower. And your favorite color, purple, is the color we associate with CF. So Maggie, welcome. Maybe tell us a little bit about yourself and tell us about, how you decided that you wanted to pursue phage therapy and made it happen when it wasn't available locally, but with the team where you're being cared for, help make that outreach with you.
Yeah, so it's kind of a sort of long story, but it all started with Emily's Entourage, which is connected to everywhere. They're a great group that does CF funding. They had pointed me in the direction of Phage back in probably it was like 2019. Um, and they gave that to me because I have MRSA, bacteria growth. which is very detrimental, obviously, for people with CF, and for some who don't have CF. So I've had a lot of trouble my junior year of high school. They pointed me towards phage. It took about 4 years for connection to Yale to happen and the FDA to approve a compassionate use for me. So it took a while to get to this point where in 2023 I started the phage, which was fantastic.
So you were having trouble with the MRSA, and your team, obviously there's a lot of there's MRSA medications, but they're not all really doing what they need to be doing, just like with Tom's case with Acinetobacter. So you heard about this therapy, and so you really made the outreach yourself. You said, I want to pursue this, and made it happen that way.
Yeah, absolutely. We reached out to Yale. They got back to us, and then it took a while through the FDA in order to get that compassionate use for me. So since then, I've been doing a nebulized form of vancomycin antibiotic, and I've been on that for 8 years now. So I started in 2023 doing the phage with the antibiotic, kind of like Steph was talking about. That's a possibility of doing, and that combo has worked really well for me. But so when I first started phage, it was totally scary and just to start something new. I think I am the first person with CF to be treated for the MRSA with the phage. So it is the first.
And inhaling it, I mean, that must have been kind of strange, to inhaling these billions of viruses.
Yeah, you walk into that doctor's office going, oh, I really hope I'm doing the right thing here, because you just don't know, if this is going to react differently to your body or to whatever. Even though my doctors were all very confident going into this, but just personal nerves. So yeah, I do the nebulized phage and the nebulized vanco. The phage only takes 15 minutes to nebulize, which was fantastic for me. It came in these tiny small vials that were really adorable. You do the needle into the vial. So it's very similar to what I do now anyway. So it's just an added treatment that I had 15 more minutes in my day to do. I did the first dose in the hospital, but then I was able to come home with it, and I did it for a week. And in the next couple of months, I see a ton of improvement. Sometimes it does take that extra couple of months just to see the full benefits of this, but I can definitely notice a difference after I've done a treatment. So right now, how they decide when I do treatments is they continuously look at my MRSA levels, and they will decide if they're high enough that I should receive phage in order to bring them back down. And again, I do that in tandem with the nebulized vancomycin. So it's all a guessing game in a way.
First of all, I was gonna just it's Andrea as our marketing person. So packaging still matters. It's a cute packaging, so that helped for your bacteriophage. But your people with CF, and I thought maybe Andrea, you can reflect on this. When you are this desperate and you're feeling so sick, and these bacteria are, inhabiting your lungs, and you're just not able to do anything about it. You'll do almost anything. And I know everyone told you it was safe, but Andrea, I don't know if how that resonates with you, and doing new things or doing whatever it takes.
Yeah, well, I'm always trying to figure out what the newest, greatest thing is. I wasn't the first person to ever transplant, but I was definitely in early days. But I have to say that, phage seems like it's come up way faster and louder than anything else that I've heard about in a while. And my question, I guess, maybe Amit, to you as a clinician, do you have a lot of people asking for it? I mean, because of all of the Pseudomonas new developments, or do people not know about it? Why isn't everybody freaking out and asking for it?
Well, I mean, so yes, people are asking, and we do refer to Stephanie's group in UC San Diego, Pittsburgh, Baylor, and also to Yale, absolutely, because Yale is close to us. But it is not FDA approved. So it's kind of a one study of one, N of one study, as we say, where we have to get approval from the FDA. So it is a process. However, people like Ben Chan, who Maggie has met, and with Dr. Koff getting treatment, They know that process and they can help with it. We will very often send slants or cultures with bacteria to these laboratories to screen their phage library for potential cocktails. These library cocktails are used in the phage lingo, and they'll get back to us. And if there is a hit, if their library is able to treat that organism, then we'll send people down. The new modulators have taken that down a notch with people a little bit healthier now and the infection's not playing as an acutely progressive role. But absolutely, there are people who need this therapy now. And I just sent a sample to Pittsburgh just 2 weeks ago for screening.
One thing that I didn't mention is that there's 2 different lifestyles that the phage undergoes, and one is what I call the phage rage kind, which is what we want, when we're treating a patient because it kills the bacteria and the baby phages
Yeah.
burst out and they go on to attack new bacteria. That's great, but sometimes, we can only find phage that are what I call the sleepy phages that enter the bacterial cell, integrate into the DNA of the bacteria, and then hit the snooze button. Well, we don't want those for
Yeah.
phage therapy because first, they don't kill the
Yeah.
bacterial cell, and second, they can actually carry genes with them that might hurt up the patient later on down the road. So, in some cases, and this is why it's important for CF patients to know this, because there's certain infections that are common in CF patients like Burkholderia cepacia or Mycobacterium abscessus or Stenotrophomonas, which I call the snuffleupagus. It's kind of one of those really long words. And those infections, we tend to only be able to find sleepy phages for them, or at least they predominate.
Mm.
And so when that happens, those phages need to be trained or genetically modified to make them the phage rage kind. And that's what was done in Isabel's case in the UK. And but we are trying. We when I say that, I mean we the phage community
Yeah.
is trying to identify, and phages that are these lytic phages or the phage rage kind to put in a registry so that they're ready, so that we don't have to go back to sewage or other environmental sources every time there's a new patient. So we need resources to be able to do that, and also the research that is being done to try to figure out how to keep a phage in the lytic phage rage stage rather than having it convert over to the other kind. That's the kind of stuff that requires some basic science research. So it isn't all clinical research. There's some basic science that needs to get done too.
And thank you for that. And I'm thinking that we're going to put a little video on the different like life cycle of a phage, because you're right that the sleepy phage is a problem, and that does explain. So thank you for that for me, because we have a lot of Burkholderia, Cepacia complex organisms, and we're often turned down as far as phage being active against them, and that might be the reason that they're not in the right state of mind. Those phage.
I'd like to suggest a rename and we call them lazy phages instead of sleepy. Sorry.
There's some
We
more
do.
marketing for you.
I'm gonna
We
steal
talk
that,
about a
lot of.
Andrea. You stole phage rage.
Lazy
I'll steal
phages.
lazy
Well,
phages. Okay, cool.
there we go. We'll be even then.
Absolutely perfect.
I have so many questions right now. So I don't have a scientific brain. It's a little hard for me to process, but I do want to go back as the non-scientific brain here to clarify something. When it comes to phage therapy, is it always like a cocktail of different phages that you treat someone with, or do some people just have like one type of phage? Because you say billions of phage over time, so I'm just trying to like understand: is it one phage over time? Is it multiple?
Yeah, okay, this is a great question. So, and I have to be honest that, I because I'm not a physician and I'm not a virologist, I'm going to put this in the simple terms that I understand too, right? We all have different levels of scientific training. So, when it comes to phage attacking bacteria, we don't know exactly the right number of phage to use because a lot of the research that we need to have done hasn't been done yet. So we're doing our best guesses. We try to use a cocktail that has different phage not just, identical phage in numerous quantity, but phage that are from, say, different families, because we want to make it difficult for the bacteria to become resistant to the phage. We know that bacteria and phage have been duking it out, having a World War III of their own at a nanoscale, right? Like for like almost like 4 billion years that's been happening, right? So what happens, like they're just using our bodies, right, as like the military tableau that they're attacking each other on. So when the phage attack the bacteria mutate, and part of that mutation comes from their immune system, and they have these things called CRISPRs, and you might have heard of CRISPR. And it's, we hear about CRISPR these days because it's genetic scissors that allow us to do gene editing, but the source of it is because that's part of what the bacteria uses to defend itself against phage. Well, phage have their anti-CRISPRs. Okay, so you can kind of think of it as a Game of Thrones type of thing, and they're constantly changing these two types of warriors. And so we want to be able to use different types of bacteriophage, ideally from different families that are attacking different receptors. And the receptors are like the door or the window that the phage is using to enter the bacterial cell. And if we're using phage that are attacking the bacteria in different ways, then there's good evidence, and some scientific studies have shown that it's harder for the bacteria to develop resistance against multiple phages at the same time. We often also use phage in concert with antibiotics because there have been cases, including my husband's case, where there was synergy between the phage and the antibiotic. So that the bacteria has to make a genetic decision when faced with both the phage and an antibiotic at the same time, and it would often rather face the antibiotic than the phage. And in my husband's case, the bacteria, dropped its slimy capsule. It was its coat, and in doing so, it lost the receptor that the phage used to enter the bacterial cell. But so the bacteriophage couldn't attack it anymore. But that wasn't a problem because it became susceptible to the antibiotic that it was resistant to earlier. So that's another tactic that we use. But we do want to have multiple like copies of these different types of phages to attack the bacteria, because there's like billions of bacterial cells in an infection that's systemic, but we don't really know what kind of dose to give the patient, right? Because we haven't done that research. But the good news is that if you give bacteriophage in sufficient quantities, and we're overdosing patients right now because we haven't seen any side effects of phage therapy. I mean, there's a couple of reports in the literature, but it's so rare. So if you're giving like a billion phages in a dose, that isn't necessarily the amount of phage that the patient is getting, because the bacteriophage are multiplying, right? Like the drug is alive, right? You can think of the bacteriophage as a drug that's alive. It's multiplying as long as there's bacteria for it to attack. So it's like the bacteriophage will die out and will be eliminated by the body if there's no more bacteria there that has the receptor that they match to. So that's the beauty of this kind of precision medicine, because in contrast to antibiotics, antibiotics have this kind of a scorched earth approach to things. We used to think that broad spectrum antibiotics that kill a lot of different bacteria was good because you didn't necessarily need to know what kind of bacterial infection it was. You just give the patient a bunch of antibiotics and it takes care of itself. Well, we know now that it's not good to kill some of the good friendly bacteria in our microbiome, right? Because it disrupts the microbiome. So this precision kind of approach that phage have to eliminate the pathogenic bacteria and leave the friendly bacteria behind is exactly what we want. And so that's one of the benefits of phage therapy.
And from the book, I remember it was 9 different phage not like the cocktail contained 9 different phage.
Yeah, but we didn't have the time to sequence the phages, so they ended up being very similar to one another, and resistance happened actually very quickly. But in Tom's case, he didn't have immunosuppression like a lot of CF patients do who were taking steroids or whose immune systems have been weakened by their disease. And so if we just knocked down the bacterial burden in his body enough that his own immune system could kick in and fight it itself. So we treated him for about a month with phage. We didn't know how long to treat him either. We just guessed. And he cleared his own infection within 3 months. Even though we'd stopped the phages kept working
Yeah.
even after we stopped giving it to him.
With phage being such an individualized type of care, do you see it becoming a standard in CF care down the road, or is it too hard to collect all the individual phages? Like
Yeah.
down the road,
Yeah.
do we see this becoming super easy to do, or will it still always be that complicated? Find the right
Well,
cocktail.
that's actually a really good question too. So basically, you're right that phages need to be matched to a specific bacteria, right? But there are some bacteria that have fewer phages required because, like MRSA or MRSA, in particular, was caused by staph, right? MRSA is methicillin-resistant Staph aureus, and it's thought that maybe 20 to 30 phages will be sufficient for covering the majority, let's say 80% or so, of staph infections around the world. And in that case, you could have a fixed cocktail that you take off the shelf, right? Rather than having to personalize it every single time to somebody. Now, in the case that my husband had, he had this pathogen called Acinetobacter baumannii. And the phages for that bacteria are really picky. They have to be matched not to just the bacterial genus and the species, but the isolate. So you might need hundreds of phages in a phage library to be able to ensure that you can match those phages to a specific bacterial isolate of Acinetobacter baumannii. But the alternative for that, rather than having a huge library for some of these infections, would be to actually genetically manipulate the phage so that they can cover a wider variety of bacteria. So there's teams around the world that are genetically modifying phage, and the biotech companies like that idea better because it's easier to patent genetically modified or synthetic phages. And so I think though, and a lot of people do think that phages are going to become a standard practice used alongside antibiotics, probably not used alone very often. But they have been used alone. In the case in the UK of Isabel, they that only a single phage was found to be the phage rage kind that we like that will kill the bacteria. So there was two other phages that were sleepy phages that needed to be manipulated and modified so that they were the phage rage kind. But since then, that one phage rage kind is called Muddy, Muddy. It's a very powerful phage. It covers a lot of different bacterial infections that are mycobacterial in nature, and it's been used by itself, and we haven't seen resistance necessarily arise, especially if a patient is immunosuppressed and may have less of an ability to mount a response in exchange for that. So the short answer is, I think years from now, it's still going to take years for this clinical trials to get done. And that's why we need this parallel track of compassionate use phage therapy to be able to ensure that patients like Maggie can get therapy. And I know that a lot of patients are waiting and they're waiting too long. And some of that is, like I said, because there's volunteer labs that are not staffed to be able to do this. And so what we need are more resources to be able to have commercial labs and commercial facilities to be able to manufacture clinical grade phage so that we're treating phage like the living antibiotics that they really are.
Now, a plug for the documentary The Good Virus that Maggie has a little cameo in, and they talk about APT, this company in Maryland that was bought by Biomex, which I believe now is bought by the CF Foundation. So we need that work, and then just we need that to continue, because it is such a promising therapy. But I also want to plug for Stephanie your book, The Perfect Predator. And when I was reading it, I found myself choking up, and there were times, especially in the beginning of the book, where a colleague said something, that was overheard. That was right in the beginning where you said, someone said, "Does Stephanie know that her like Tom is going to die?" And then they didn't realize that they hadn't hung up. And then other times where you needed to, sort of take charge. And if you hadn't, I mean, Tom, like really, I mean, whether you did use bacteriophage like later, getting him to that point, you needed to advocate. And I think that resonates with a lot of people with CF, that having someone there to advocate for you and to just constantly be that support. And I know, Andrea, that's something we've talked about, and we have a caregiver episode on that. But tell us about that emotion. I mean, it's such incredible work and energy and emotion that like you have to expend to take care of Tom in those
Well,
days.
thank you for raising that because it is a really important issue for the CF community. And we were in 3 different healthcare systems, Egypt, Germany, and the US. And I had to advocate in each one of them in different ways. So navigating the healthcare system and standing up and asking questions and getting educated is one of the most important things that you can do as a caregiver, regardless of what, the infection or disease is. But I also, I mean, that's one thing I've really been impressed with, the parents of patients with CF that I've come to meet, and CF patients themselves are so, like, resilient and strong, and the things that you folks have to go through, you're my heroes in so many ways. But I also wanna acknowledge that there's so many different CF foundations and institutes. Maggie mentioned Emily's Entourage. Emily's Entourage has funded an MRSA phage library that we've been working on at San Diego at IPATH. The CF Research Institute and the CF Foundation have funded us and other programs around the US. And without that advocacy at an institutional level, we wouldn't exist. It's hard enough as it is right now. So, people with big pocketbooks, please donate. I donate my time to IPATH. I haven't been paid by IPATH or by the university for the work that I do for IPATH in years, but that doesn't matter because I feel like I had the gift of my husband's life when all else was almost gone. So I'm here for you guys. I answer every email I get. I don't always have the answer, but I'm still getting educated. But we're gonna do this. We're gonna make this happen.
Stephanie, you're really incredible, and you do answer those emails. And you're right. I'm so glad that you mentioned the CF Foundation is huge, and the biotech industry is they're assisting with these projects. But CFRI and Emily's Entourage, they're funding those projects that are leading the biopharmaceutical companies to get the courage to take these things on. So, really appreciate that. Andrea, Lauren, I would love to have Maggie just say a few other words, but I want to see if you guys had any questions.
Maggie, how do you feel?
I feel good. Honestly, I've been doing really great the last couple of years, so I do attribute that to the phage. And right now, I'm also feeling an awe meeting like Stephanie because your work is obviously what's helped me now.
Well, I have something I want to say to you, Maggie. First, happy birthday. Okay, it's May
Thank
is
you.
CF month, as
Yeah.
you've learned, and it's my birthday this month too. I'm turning 60, so we're
Oh, wow.
twins, I guess. And
Happy
Yeah,
birthday to
and
both of us.
it takes courage to stand up and to share your experience. It took courage for, Tom and I to write our book, and so I know how vulnerable it can make you feel to do that. So, people like you and Ala Balasa and others that have spoken up about their experience, it really does inspire a lot of people like Lauren that are, just learning about this and might feel like, wow, this is something that I might want to consider or that I might need someday. And so thank you for doing that, especially at such a young age to have such poise and maturity. You're really awesome.
Thank you so much. That means a lot. I figure whatever information can get out into the CF community will help anybody at some point. For instance, this is a connection I made earlier in the podcast as I was listening to it. Mallory, you were talking about, who was the incredible woman with CF, who first started the phage therapies with her. I am taking a phage called Malokai, which I believe stems from Mallory's exploration into phage. I think that was the first one she created, and I think that's named after her, if I'm getting my facts correct. So it's a revolving door that it's so meaningful and deep that eventually stuff happens, and we have hope with CF now, and that there are things in the pipeline just like this phage. Because I'm also not available to get any of the modulators at all yet, so I'm still looking for the next step, which is this. So it's really incredible.
So acute chronic infection is still something that's like very prominent part of your life, Maggie. And so absolutely these therapies, I mean, for those who aren't on modulators, so critical, but even for those who are on modulators, and then globally, I mean, people with CF are suffering without those treatments and, and dealing with infections. And we're, in that documentary we watched, that work being done in Africa, but just it's so critical to get this, make this available globally. And thank you for both of you. I know we're running out of time, so I think we're going to have to pivot. And there's so many. I know, Lauren, you have like 1,000 questions. I have 1,000 questions.
We're going to just have to do another episode
Yeah.
with you guys.
We talk about a lot of heavy stuff on the show, but we always like to laugh a little bit. We're going to do a very quick lightning round for a little bit of fun to end the show. And Amit and Lauren are going to ask you guys simple yes or no, true or false questions. You guys ready to start?
Yep.
Stephanie, do you hit snooze or get up immediately?
I get up immediately.
I knew it. Maggie?
I snooze all the live one day. It's so bad.
You and me both. Okay, teleport or time travel?
Teleport.
Maggie?
Ooh, probably teleport. I think I like traveling, so.
Dine out or cook at home?
Dine out.
Cook at home.
Okay, unlimited travel or unlimited free food?
Unlimited travel.
Can I, give the free food to everybody else on the planet? Because some will hunger that way?
Aw,
It's just for you.
No, that's such a
is.
great
Mhm.
idea.
I'll still
Phone
take
on
free-
silent or volume on?
Volume on.
Volume on.
See, I'm a silent girl. Visit the jungle or visit the desert?
Jungle.
Jungle.
Are you the first to arrive or last to
First
leave?
to arrive.
Last to leave.
Save leftovers or throw them out?
Save.
Save.
Right. Watch movies at home or go to the theater?
Home.
Home, unfortunately.
Do you prefer texting or talking on the phone?
Talking on the phone.
I'm a texter. I'm learning phone conversations, but I'm a texter.
Gen Z.
All right.
I
the
know,
last one,
exactly.
it is so generational. Okay, this is the last one, and your go-to road trip album. I mean, when we did this, but what is your either a song or an album?
Oh, mine would be Road Apples by the Tragically Hip. And a road apple actually is a big, horse plop of shit. So that's very Phagey of me, right?
It's so appropriate. Yeah.
Oh my
Nice.
You're on brand.
Right. Oh, this is a tough one for me. I've been on an ABBA kick lately, so maybe ABBA. But also Zach Brown Band. My dad has a CD playing over and over and over, so maybe
Aw,
that.
I love that. Very nice.
Okay, that's our game. So now is the point in the show where we ask you guys to sing our theme song ending. And who would like to go first?
Oh, I will. What the heck? Okay. It's a long story.
Woo!
That was
Wow.
amazing.
With the hands. I love
Yeah,
like
I know.
jazz
Jazz
hands.
hands. Jinx.
Okay.
Okay, Maggie.
So
You got this.
Okay. It's a long story.
Nice.
Really
Beautiful.
nice.
Beautiful.
All
Oh,
right.
I love that.
All right. Well, with that, until next time,
Get a little older.
Live a little bolder.
we are working hard to provide meaningful information to a deserving CF community, information you won't find anywhere else. If you'd like to help us grow, we'd greatly appreciate it if you would follow us on Apple Podcasts, Spotify, or wherever you get your podcasts, so you never miss an episode. Want to go all in? Leave us a rating and a review. It helps people find us and makes a big difference. If you found this episode helpful, please share it with a friend, family member, caregiver, or provider who might also want to hear it. This helps you, it helps them, and it helps us spread the word to those who need to hear it most. It's a Long Story is produced by me, Andrea Gavin Becker, and is hosted by Dr. Amit Euler and Lauren Harvey. Additional editing and production support is provided by the team at Resonate Recordings. Until next time,
Get a little older.
Live a little bolder.
It's a long story.
Resonate.